Okay, welcome back. I'm quite humbled as a little workshop organizer to introduce one of the program organizers, Rudolf Leube from Aachen. And another special thing to note is so that Rudolf actually has a medical background. It's not only biologists and mathematicians and physicists here in the room, and we move on to the intermediate filament system.
Till?
Yes.
Could I ask you to ask anyone who asks questions to speak up? Because if you're at the front and you ask the speaker a question, the people at the rear can generally not hear them.
Or maybe the speaker could repeat the question.
Yeah.
Okay. Yeah.
Okay.
All agreed.
This may be a challenge because I'm sometimes hard of hearing, and I had to fiddle with my laptop to get it going, and I had some coffee, but I think everything is set up now, and I hope I will be able to give you first a short introduction to what intermediate filaments are, and then sort of summarize our work from in vitro to in vivo, and trying to show you what we quarrel with in the laboratory. Now, if you think about intermediate filaments, all right, this is the first trick. You have to put it on. It's probably the part of the cytoskeleton that is most enigmatic and has been neglected for long times. So they coexist with the other two major filament systems that's based on turbulin and on actin, and in terms of quantity, they equal the other systems quite a bit, especially in epithelia. Now, when we talk about intermediate filament systems, it is a very diverse group in terms of composition. And that's probably what deterred lots of researchers to even work on these type of diverse molecules. So you have a list here, and you see more than 70 polypeptides that can form intermediate filaments, including those that are located in the cytoplasm, and those that are localized in the nucleus, and those that have specific functions in certain tissues. And then on the basis of their assembly properties, on the basis of their gene organization, and on the basis of their primary amino acid sequence, they have been grouped into certain types, type 1 through type 5, or this is sometimes referred as to type 6. And when you look at the different polypeptides, is a big zoo of different names. What is nice for the medical community is that they are so diverse, because they can be used as very specific differentiation markers of a given cell. And on the contrary, you can also argue that probably they have adapted to very cell type specific functions, and that's why they're still evolutionary quite active. And if you look at evolution, for example, of epithelial intermediate filament polypeptides, we have identified genes that have been inactivated during the last 1 million years between primate and human development. Now, the most complex groups are those of intermediate cells are those that are found in epithelial cells, and those cells that form the hair. And they are grouped in type 1, 28 members, and type 2, 26 members. So since epithelial tumors, that's cancers, are derived from epithelia, they have found a proper place in cancer diagnosis as very reliable differentiation markers. What's important to keep in mind is that keratin intermediate filaments are obligatory heteropolymers consisting of stoichiometrically actually even amounts of type 2 and type 2 polypeptides is expressed in these cell type specific fashions. Now, the last, I'd say, decade or so has witnessed that actually these very abundant polypeptides in epithelia contribute to various epithelial functions, although they're not essential for these epithelial functions, contributing to migration, proliferation, differentiation, and even such complex processes such as inflammation. And conceptually, we think about these very abundant polypeptides in epithelial cells as an incredible buffer repertoire that can act to protect epithelial cells from various types of stress, which not only includes mere mechanical stress, but also UV stress, chemical stress, or even microbiological assault by certain organisms. And what's also remarkable about these diverse polypeptides is that they've been associated with a large number of disease. Most of them are genetic diseases, but that also includes more common diseases, such as predisposition for certain organ-specific diseases, or again, being involved in tumor formation. And the way we look at it is that you have this abundant network with this very large surface that forms cell type specific distribution patterns, either underneath the plasma membrane or throughout the cytoplasm. And if you and this can act in one way as a stress product, as a buffer system in the cell. And if you have mutations in these keratins, this buffer reservoir or this stress protective activity is sort of limited. So if you then induce metabolic stress or mechanical stress, the network organization will deteriorate into granular aggregates, which in liver disease are very well known as Mallory Denk bodies, one of the most prominent signs of alcoholic liver disease in terms of morphology, or in skin diseases, you find multiple granular aggregates in the cytoplasm instead of this very nice three-dimensional network. So in a way, we consider these filament systems as being essential for epithelial homeostasis, but not essential for epithelial formation by itself. In trying to understand what these filaments and their specific organization do, I go a step back, is to assembly of these polypeptides in vitro. And there are some specifics that distinguish them from the other filament systems. So the polypeptides that make up these filaments are not globular shaped, like in the case of actin filaments and microtubules, but are rod-shaped. And that's actually the main feature of these polypeptides, as all of them share, is a rod-shaped domain consisting of a little bit more than 300 amino acids. And these 300 amino acids form an alpha helical coil domain, and these alpha helices contain hydrophobic residues on the surface which allow very stable dimer formation. And these dimers are so stable that you need very high concentrations of chaotropic substances, for example urea or guanidinium hydrochloride, to actually dissociate these stable heterodimers. What is not drawn in this image is that these heterodimers here show only this central rod domain, the part that is common to all intermediate filaments, at least from a structural point of view. But you have head and tail domains, which are depicted here, which can be of very different size, very different composition, probably very different types of structural composition. So these are the parts of the molecules that make them specific. So they must confer some sort of cell type specificity, and they're also subject to post-translational modification and probably modify this assembly behavior. Now, if you look at the heterodimer, it's always aligned in a parallel fashion, whereas the next sort of assembly step that we can observe in vitro is an anti-parallel association. So what happens in this case now is that the originally symmetric molecule with an N and carboxy terminus, this subunit now, you can flip it either way. So it doesn't have an intrinsic symmetry like you would have with the other filament system. So these polypeptides are intrinsically non-polar. And that's the message of this tetramer, which most likely is the part of the molecule which exists in the cell in the cytoplasm, because it forms spontaneously. And that's the other message I should like to convey here, that these assembly processes and even the further steps that I will show you very soon occur in the absence of any cofactors. So you don't need energy, ATP, GTP, as the case with other filament systems, and you don't need assisting molecules. That occurs spontaneously. We just have the right pH and soil conditions in in a liquid, just in a buffer. So the next assembly step, and now we get into sort of dark and not so sure area, is the formation of a so-called unit length filament. So you have lateral association of 6 to 8 of such tetramers forming a 60 nanometer long rodlet that's referred to as the unit length filament. And these unit length filaments, with their ends sticking out, will then longitudinally, by interdigitating their ends, form the mature 10 nanometer filament. And actually that's what these filaments are also referred to as 10 nanometer filaments, which form these sort of endless filamentous structures in vitro.
Rudolf, sorry for yelling at you, but they want to hear the question. What happens to the head and tail domains in these thicker filaments? There must be a huge place constraint on them.
Well, most people think that they stick out of the filament as sort of little dot structures, but it's not resolved if there's also inside or not, and where they're placed. But that's a very good question. Yes?
I don't understand. The building block is 60 nanometers, and then you say a 10 nanometer filament.
What is? Oh, sorry. This is the length, and this is the diameter. Thank you. Thank you for the clarification. Okay. So let me just point out again the basic features. We are talking about non-polar structures. They assemble spontaneously in vitro, and but I didn't say they are very major phosphoproteins in the cell, actually also subject to various other post-translational modifications. So what we still don't know, and I don't have an answer to you for that, how if they spontaneously assemble, how is their turnover regulated in the cell? I mean, must be dynamic, must be flexible. If they're non-polar, how do they know where to start from and where to end? And how is this assembly procedure regulated in a really a cell? And that's probably through post-translational modification. And it was about 15 years ago that we started work on this in trying to examine how these processes operate in a cell. And at that point, our view was that you have this stable network in a given cell that just stays in the cell and is rigid and provides mechanical resilience. Now, with the use of fluorescent polypeptides, we could still look at these cells at different time points. If you just compare these time points by themselves, you see the network organization sort of seems overall the same, but it changes dramatically from here to there. And if you then animate the sequence, you actually see that in the cultured cell, these filament networks are not static, but they are highly motile networks as depicted in this timeless fluorescence movie.
Can I just ask, in that, have you got GFP or something on it?
Yes.
Because doesn't that disrupt the ability to polymerize?
That's a good question, and it probably does affect it. I mean, any kind of label you introduce to any given molecule will definitely have some influence on it. But we were very careful to use stable transfected cell lines. We compared it to the non-transfected cells. We looked at the filament distribution is the same, and basically it's quite the same. And maybe I should say this already now, but at the end of the talk I will show you that we can use these labeled polypeptides and substitute the endogenous gene with them and have no functional deficits. So I think it's a pretty good argument that it's very similar, but it's clearly not the same, so especially if you talk about kinetics and things like that.
A question?
Yes?
Are these filaments cross-linked, or is the apparent cross-linking in the movie artifact of the representation?
Well, it probably depends on which keratins you look at. There's evidence that there are cross-links, disulfide-mediated cross-links, and that there are linker molecules such as plectin. But most of the things you see here is just because they're projection images and not true cross-links, because the filaments themselves are endless and they don't have a specific side connection to each other. Yeah. And to illustrate this a little bit further, the dynamics, and I will probably go over these images kind of quickly. I show you one experiment here, in which you have a control cell here on the left, and this cell here we used fluorescence recovery after photobleaching, by which you bleach one half of the cell, and you look at the reappearance of fluorescence. And what's remarkable about the reappearance of the fluorescence is, first of all, at the end of the several hour time movie, the fluorescence again distributed throughout the cell rather evenly. And the reappearance of fluorescence is not equally to each part, but seems to occur in a directed fashion. So if you look at it, the fluorescence will first appear from the cell periphery. You see sort of more fluorescent filaments moving inwards, and only at the very end you will have reappearance of fluorescence in this area that is located around the nucleus. So we have, or we postulate from these and similar experiments, that we have a spatially defined turnover of the keratin system in these cells. And in the next few slides, I will summarize work that's actually taking us several years, by which we describe the different steps starting from the cell periphery to the cell interior that we could distinguish. So what we see in the cell periphery at higher temporal and spatial resolution is that close to the plasma membrane, which would be located here, you have an appearance of little small particles. These little small particles appear there. We refer to this process as nucleation. These particles grow longitudinally, a process that we refer to as elongation, and it just shows growth of one such particle. Here are several particles in this composite diagram. And these elongating particles then fuse with each other and integrate through their ends into the peripheral network. This process that starts here in the cell periphery is independent of protein biosynthesis. So if you treat the cells with an inhibitor of protein biosynthesis, you still have this cycle ongoing, which means the cycle is fed from pre-existing material, which already was suggested from the photobleaching experiment. Now, if you look at these particles, they seem to come from similar areas, and the motility of these particles is directed. It's not just stochastic movement of these particles.
So question, how does this picture change in a polarized cell? Let's say I have a polarized cell with a lamellipodium on the front, some contractile part in the back, and then the sides maybe have some fibers or whatever. Where would the...
I can make predictions. The proper measurements will be made. In a polarized cells, we would expect that we have increased nucleation in the leading part of the cell, and maybe reduced formation of particles in the trailing part of the cell.
But wouldn't it interfere with the actin meshwork?
In which way?
So the lamellipodium needs space, it's very dense.
Well, they have space there.
It will be in front of all this.
Okay.
I'll show you some movies for that in a bit, but maybe. It did work before. See? No, it does. No.
Kind of.
No. Let's use this here. So this one is not running for some reason. So here we have a cell with very prominent focal adhesions labeled in red. You have the filaments here, and this shows you one such focal adhesion at much higher magnification. And what we see that in close proximity to these focal adhesions, you find these nucleating and elongating particles which fuse to each other. And that focal adhesions actually play a role in this is suggested by the observations. If you disrupt the focal adhesions, you will find reduced formation of these particles. But at this point, and we've really tried for many years already now, we really don't know what the molecular mechanisms might be behind that. We know what is not. We have tried some of the prominent candidates, but we really don't know what drives the formation there.
This answers my question. So the lamellipodium will be, of course, before the focal adhesions. And
Yes. Oh, yes.
behind
I come to that too. Yeah. So we are talking about the lamellum. So it originates as a lamellopodium-lamellum interface. Yeah.
So these things are just very thick bundles because you talked about this 10 nanometer.
No,
before.
they're not thick bundles. They're probably single filaments.
But how can you see them on this length scale?
Because it's fluorescence. Fluorescence doesn't tell you anything about the size of a filament. It does not.
You think each of these is a 10 nanometer thick?
That's the best I can guess. I mean, I cannot exclude, and you have to image it to really show it's truly a 10 nanometer filament, but I don't have any indication.
It's just compared to what I know from actin, which has roughly that same thickness, a little bit thinner, right? But it's completely impossible to see individual actin filaments on a.
Why shouldn't it be possible? If you have fluorescence, you can see much smaller. It depends on the labeling density whether you can pick up the fluorescence signal.
A quick question about the attraction and repulsion of these fibers and the elongation. Is there a repulsion between fibers? Because what stops them all just bunching together and making a mush? What keeps them as separate fibers while they're elongating?
Well, I would rather think about the other way around. How do they find each other? To me, this is a miracle.
And it's just noise.
If I look at the space, it's a miracle how they find each other, the ends. Because you have very few particles. There's not that many. How do they find these ends that have to interdigitate? It's really, I think I would really rephrase it exactly the other way around, the question. Okay, maybe it will become a bit clear in the next few movies. So we're wondering about what drives these particles here. And here you have a double labeling with, again, the keratins with the actin stress fibers, and you see that these particles in most parts are transported along these actin filaments in a comparatively slow kind of continuous motion. But look at this guy here, along these fibers, and it's inward directed. But then you also see occasionally, and this was the one I think I pointed out to you, which particles that move very, very rapidly. So this kind of motility has the signature phenotype of an actin-dependent type of motility. Yeah, see again up here. So we can actually uncover this kind of motility even better if we destroy in a cell the microtubules, the cytoskeleton collapses, and you find then that these particles or even the filaments reorient along actin stress fibers, and you see that the motility of the particles continues in this rather slow, continuous, and inward directed fashion. But conversely, if you treat cells with a drug that disrupts the actin filaments, in this case we use letrantolin, you again find a collapse of the network, and now you see the particles out there, they're not static. They still move, and now they show the signature phenotype of a different type of motility. It's much, much faster, almost 2 orders of magnitude faster. It's not continuous. It is a stop-and-go kind of motility, and it's bidirectional, which identifies as a microtubule-dependent motility. So putting this together, these particles can latch on to either one of the other filament systems and use them as tracks for motion. So how do we think this whole thing is working? And I'm using here a scheme that we published a number of years ago, in which you have the peripheral keratin network, you have the microtubules, and then you may have a preceding lamellopodium with a lamello. So this is a little bit too close to the plasma membrane. I agree. You have here a nascent focal adhesion which matures and then uses as a attachment site for actin stress fibers, and then some react this way. I don't know the molecular mechanism for this. You find nucleation of keratin particles in the near vicinity. They elongate, fuse to each other, and become integrated into the peripheral network. Now what happens next after these particles have been integrated into the network? The network itself continues to move toward the nucleus, And if you track individual filaments, this is the cell periphery, this would be the nuclear area here, you find that these filaments, which are low in fluorescence intensity, become more and more fluorescence, indicative of bundling behavior that we believe is due to association of several 10 nanometer filaments in this process that we refer to as bundling, which is driven by, as I said already, intrinsic properties of the molecules, those sulfide bondings, but also by extrinsic mechanisms, that is by cross-linking through flectin. And if we now follow these filaments that become brighter and brighter, you see another phenomenon. Here's the nucleus now, in this case here. These filaments lose fluorescence again as they go closer to the nucleus. And if actually do this quantitatively, you see that these inward moving filaments lose the fluorescence, while those are used as a control here in the nucleus or in other areas of the cell remain fluorescent here at 100%, as shown here. And we believe that this loss of fluorescence is due to loss of subunits in a process that does not generate little particles, but just releases the tetramer subunits. And this is another experiment that supports this idea, in which we, by photoactivation, made half of the cell fluorescent. And after here several hours, more than 3 hours, you see that the fluorescence is distributed throughout the cell. The source of which can only and exclusively be those fluorescent filament that we labeled in this here sort of pulse chase experiment, showing that there must be some kind of exchange from this part of the cell to this part of the cell. And we can actually also measure the diffusion coefficient of the soluble material in these cells by various microscopic means. This is one part. So filaments disassemble into soluble subunits, but there are also filaments like these here that are around the nucleus that are extremely stable for several hours. I think this is a movie of close to 8 hours, and just remain the same. They don't lose their fluorescence. And similarly, filaments that are anchored to desmosomes seem to be quite stable, and those that are anchored to the attachment sites to the extracellular matrix on the bottom of the cells also appear to be quite stable, in contrast to the peripheral domains of these still very highly active cells, as shown as by these lamellipodial protrusions. So we put all these data together in a scheme that we refer to as the keratin cycle, which is fed by diffusible subunits, or also subunits that are produced through biosynthesis, of course, which will nucleate preferentially in the cell periphery close to focal adhesion sites. These particles will elongate into little filamentous structures which fuse, integrate in the peripheral cytoskeleton, which bundles and is continuous move from the cell periphery to the cell interior, mostly in actin-dependent fashion, and then either the filaments disassemble and sort of go again through the cycle, or become stabilized through a maturation process as anchorage to the nucleus or to various cell-cell adhesion sites. And the biological relevance of this cycle, that we believe that this continuous ongoing cycle allows the cell to very rapidly, that is without protein biosynthesis, adapt shape changes and various other changes that occur in cells.
Sorry, can I just ask, this implies there must be chaperones, right? Because they're hydrophobic, so are there chaperones that take them off and allow them to reassemble some of those?
I think the question is perfectly phrased, but I don't have the answer there. If they spontaneously assemble, there must be a mechanism to prevent assembly, and the most likely candidates would be some sort of chaperones, and there's actually only one paper around that which suggests that there's such an association true. 2 years ago published in PNS, which shows that actually there's a complex between 14-3 proteins and actin and G-actin and keratin, so that would be a potential candidate. And there's some work around which also shows there's association with chaperones, but it's still very diffuse, the literature that's available on the topic, yeah, but quite right, yeah. A way to check that this cycling is actually true is depicted here. And as all of you know, if you look at a cell in culture, each cell looks very different. And you will find very different kind of behavior. And especially if you talk about kinetics in a given cell, it's almost impossible to compare one cell to another cell. So we actually spent quite some time together with Institute for Computer Vision in Aachen to make or to produce culture conditions and image analysis routines which would allow us to determine motility of these filaments and also turnover of these filaments. And the example shown here just gives you the results that we got, by which we sort of normalized the cells, put many of these cells on top of each other, from short time-lapse images of just 15 minutes. And what you actually see very nicely is that you have in this heat map the highest dynamics of the filament system in the peripheral cytoplasm. And if you depict the same here as a vector diagram, you see that the net movement of all these fluorescence is from the cell exterior to the cell interior and kind of stops in the perinuclear region. And you can use this information that you have from the time-lapse images if you look at the fluorescence intensity to then also determine the bulk flow in the cell. And the bulk flow analysis then gives you regions of assembly, which we refer to as sources, and net disassembly, which would be sinks. And very well goes with the scheme that I showed you before. The sources are in the cell periphery, the sinks are primarily in the perinuclear region. So I think this is a pretty good sort of objective demonstration that this cycle actually exists. And this cycle not only exists, but is subject to positive or negative regulation. Negative regulation is shown here, where we compared this set of cells, which was imaged just a day after putting it in culture, with cells that were put into culture after 2 days. And you see at these late time points that the motility is reduced, and also then accordingly turnover rate is reduced. And conversely, we can induce the activity by treating cells with a growth factor, in this case epidermal growth factor, by which we can upregulate the motility and also upregulate the turnover. And we could also show that this correlates with phosphorylation of the keratin polypeptides in this system. This is slow today. Now, what may go wrong with the cycle? And this shows you a pair of cells where we introduced mutant keratins, so that occur in these blistering diseases I showed you at the very beginning. And what you see here, you still have a sort of a residual, not very nice perinuclear filament network, and you see these roundish particles in the cell periphery that move very rapidly, originating from the red structures, again, focal adhesions. So yes, we see nucleation in the cell periphery, we see formation of particles, but not of really nice filaments. So what we think is happening here is that in the cell periphery you still have nucleation, but then these mutant polypeptides don't properly or not very efficiently elongate to only form this kind of odd networks, and these granules then go in a much faster turnover cycle. They actually only have a lifetime of less than 15 minutes and nucleate again. So what you're looking here at is a disturbed or perturbed capacity of these mutant polypeptides to form filaments in these cultured cells. And just two more examples, and I will not stick too much with this. If we look at during the cell cycle, here in this cell you have an interface cell which goes through mitosis, you get kind of an appreciation how rapid this entire network can be disrupted and broken down into little granular punctate material, which then, at the end of mitosis, will form a de novo network. And this is most likely regulated by phosphorylation, as a number of in vitro experiments suggest, such as this one here, in which we treated cells with a tyrosine phosphatase inhibitor. And let's see if this runs again. And you see, or you should be able to see that within a few minutes, actually a few seconds, if you're lucky, you find a disruption of the filaments into little rodlets, in this case.
I was wondering if you had a way to recognize phosphorylation patterns, and if indeed you'd seen a gradient between the periphery of the cell.
That would be very nice. So far we have not seen it using all the antibodies that have been available. I don't know of any publication that has shown that.
What's the experimental difficulty of fishing out those chaperones that Ami's talking about? That field is being plagued with exactly that problem of not finding keratin-specific binding proteins, and that'd be just one of them. I'm sure the chaperones, nothing in biology is left to spontaneous assembly or disassembly. You're gonna, I guarantee you, find multiple chaperones that enhance or catalyze or regulate disassembly and assembly and transport and everything else under the sun. What's been the problem experimentally? Why can't you just take your keratin as a bait and just fish out your protein?
Okay, I agree. And the problem is the complexity of a given keratin cytoskeleton in a cell. Usually you don't have just a single keratin, you have at least a pair, and usually you have multiple of those that have multiple phosphorylation sites and have multiple possibilities to associate with proteins. And these associations are apparently not very strong, and people have done co-immunoprecipitation experiments and all this, but have not come up with very good candidates. And I think a part of the reason why this has been so unsuccessful in the past is that we very often use not very clean systems. We now have a system in which we, from Thomas Magin in Leipzig, who has actually deleted all the keratins from a keratinocyte, and now you can restitute the filament system with just putting in one keratin polypeptide, and that I think may give us a much better handle on what these mechanisms might be and what these interacting proteins might be. But yes, it is at the end of the day, it's a riddle still in the field. Right? Quite true. But what I'd like to use the next or last 20 minutes is actually not so much thinking about this situation of cultured cells, which also is a very artifactual situation. So transformed cells, they are stimulated cells, and that's why you have this rapid cycling probably. And if you think of an in vivo situation, maybe this only reflects the situation of a keratinocyte upon wounding, or when it becomes reactivated during invasion. But in a given static tissue, this might be a very different kind of picture. You may have very stable filaments that are attached, I'm sorry. This is not the date of the that are attached to desmosomes, hemidesmosomes here, and the nucleus. But before I go to this, I just show you, since this is about cell migration, one image series which kind of suggests the polarization that I've been asked for before here. A wounding assay, we see the highest activity of dynamics actually at the leading front here, and it's made or seems to be coordinated in a way by the focal adhesions, which become more localized to the leading edge of the cells and removed from the trailing edge of the cells. Now, to examine properties of the keratin system in a more physiological environment, so to say, we made use of mice and also of a model organism, C. elegans, which we use, in which we introduced reporters. In this case, in the mouse, we actually replaced the endogenous gene for one of the keratins, keratin 8 in this example, by a YFP labeled protein that's encoded by the endogenous gene now that's been modified. And this is just one example here of a tissue that is the intestine, and intestine contains this multiple finger-like protrusion that we refer to as microvilli. And if you go to the microvilli from top to bottom, you see that the fluorescence actually is concentrated at the lateral plasma membrane, and actually this is always just if you become this here, just shortly before you reach the tip of the cell, you find this enrichment of material which is localized here as shown by electron microscopy under the apical plasma membrane, and there's this sort of dark area here where the intermediate filaments are localized, and together with actin filaments, other cross-linking proteins are involved in anchorage of these multiple microvilli, which together form the brush border of the intestine. A very similar structure actually can be found in C. elegans, and you have the anterior part of the animal here. The posterior part actually goes much longer in this way. In the anterior part you have the pharynx, and here you have intermediate filaments which show the stripe pattern, which I'm not going to be talking about. And here you find the beginning of the intestine. This would be the outline of the cell, and this is the apical part of these cells that form a single tube that consists of 20 cells and not more, and surrounds it like a glove from the anterior all the way to the posterior part. And we know that these intermediate filaments are also localized exactly in this dark, dense structure underneath the apical plasma membrane. You have the microvilli, and is anchored, similar to this situation here, to cell-cell adhesion sites that in C. elegans are referred to as the C. elegans apical junction. So we do have conserved distribution of these filaments in functionally comparable tissues, but I should stress this, the polypeptides don't show sequence homology, just structural homology, again the central coil domain in these animals. Now, if we look at the C. elegans again, I have another image here. This again, anterior posterior part. This is already the part I showed you. It's nothing more than an eating machine that has to reproduce and actually is able to release multiples of its body mass per day in terms of embryos. And it feeds continuously in a very regular cycle. So the feeding tube starts here. You have the foregut here, and this is the intestine. The 20 cells go from all the way here to the hind gut, and these are the 20 cells. And this is the violet kind of part here. And these 20 cells are depicted here by labeling to the cell-cell junctions where the filament system is actually anchored to. And you see that this filament system here is attached to these junctions that are marked in red. And if you look in the electron microscope, you see, that's probably difficult to see in the back, a dark line that surrounds this ellipsoid lumen of the intestine. Here are the multiple microvilli, and the dark line corresponds to the localization of the intermediate filaments, which are attached to the junctions that are labeled here by an arrow, either here or here on top. This just shows you in a little bit more detail how this structure looks like. Again, you have the tube, which is not round but ellipsoid, and you have the cell junctions that are marked here, and these cells forming these tubes are arranged in these ring-like fashions, and underneath here you have intermediate filaments, and here you have the microvilli and the actin filaments that are anchored to this structure here, which then attaches it to the C. elegans apical junction, and the same thing is depicted here in the animation. And here again, electron micrograph, microvilli, this electron dense junction, which, structure which is referred to in C. elegans as the endotube, and here's the C. elegans apical junction. Now, what's nice about this, we can use this very peculiar arrangement of the intermediate filaments to search for organizers by doing simple genetic screens. And this just showed you two such examples. Here's the wild type again, just the intestine from anterior to posterior, and here you see a mutant in which you find these protrusions, which we refer to as the bubble phenotype, or in this case, you have disruption of this glove-like structure, and now you find multiple lines that actually co-distribute with the junction in these animals. We actually identified the responsible genes. In this case, it was a MAP kinase. In this case, it is an organizer specific to C. elegans. I will be talking a little bit more about this organizer. What you can see here, again, the wild type, and in this mutant, where this organizer is missing, where we lose this dense structure underneath the microvilli. The microvilli kind of become disordered. The lumen, the nice ellipsoid shape, is also altered, and this electron-dense material then aggregates here, as depicted here, and also here next to the junction. So it's, so to say, a functional or structural knockout of intermediate filaments in these intestines of the worm. So we have these structural alterations, but we can also use this system to correlate this with functional disturbances and perturbations. So what we see is that this has severe implications for body size, apparently reduced uptake of food, reduction in brood size, and also, very recently know that cholesterol uptake is perturbed. And to come back to my original theme of buffer or stress protection, we can use such a system to challenge it. So this experiment just very schematically summarizes a large number of experiments in which we subjected the worms to pore-forming toxins. And as you can see, the more perturbed this arrangement of the subapical intermediate filament system is, the more phenotypic alterations do we observe. For example, increased larval lethality, development is slowed, we see sterility, and also decreased lifespan. Such genetic systems can also help to sort various factors. So we can determine what is determining which is determining which. So this mutant, this intestinal filament organizer, is important for apical localization of the filaments, but can also ask the reverse question: if we deplete the intermediate filament, how is this intermediate filament organizer perturbed? And it's not. So it argues it's upstream as an organizer of the intermediate filament system. Or we can also look through time. So if you're interested in gut formation, you see it here during embryonic stages, during morphogenesis, that you first see this filament organizer in this part of the worm, in the anterior part, where the intermediate filament system is not present, and it always precedes a little bit the localization of the intermediate filament. So we can sort of order different components in this system, and we can do this by doing genetic screens for not only localization, as I showed you, but also for mutations which prevent this mislocalization or which enhances mislocalization. And what this describes here, that we can actually you ask all sorts of also functional questions. So how is the system involved in cell-cell adhesion in the intestine? Because it's a continuous system. You have the junctions, you have the apical filament system. And what you can see there is a summary of a large number of experiments in which we did double mutations and single mutations. So what we found is, if you take either one of these various components out, adhesion is not perturbed. But if you combine components of system 1 and system 2, we find perturbation of cell-cell adhesion in these instances here. So it's really a nice system to play with the components that contribute to these systems. And finally, we can also correlate these genetic interactions to functional properties. And this is an experiment that we've done together with Rudolf Merkel, in which Oliver Janel actually prepared the intestines from the worms. It's very simple. You just make an incision, and it has a hydroskeleton, so the intestine gets kind of squirted out. And if you use the right conditions, you can actually keep the intestine vital. And this shows a micropipette experiment, which actually applies quite large forces to this prepared intestine. This is still the granite that also came out, and here you have the rest of the worm. And if you do this experiment here, you can see that it's very resilient, that you can do this repeatedly, and after such elongations, you will not quite go back to the initial length, but it will recoil. So we do have, and we can now quantify this and correlate this with morphology within the worm, properties of mechanical properties of these intestines that are repeatedly stretched, and correlate this then, of course, also with perturbation through genetic modification that we can do in the worm. And the importance of the intermediate filament system for mechanical properties also is summarized in this experiment. Now I'll go back to the cultured system, also work we've done together with Rudolf Merkel's group. And just shows you one example in which we use a magnet and a magnetic particle that's been taken up by these cells, and we measure the displacements of these magnetic beads, and after appropriate calibration, you can then see that actually depletion of the keratins in these cultured cells, this is the keratin-free keratinocytes as alluded to before, that you have very different or reduced stiffness of the cell, and you see these particles actually in the end kind of go to the magnet here. This is the wild type, this is the mutant, and we can actually rescue this phenotype by introducing intermediate filaments into these cells, in this case keratin intermediate filaments. I see I'm running out of time, but I would like to end with a few images of in vivo observations of keratin dynamics in arguably a living mammalian organism, and this is the mouse embryo. And the mouse embryo, here's the blastocyst stage, consists of a trough ectoderm, this outer layer, and here's the embryoblast. And you see in this outer layer that produced from these labeled endogenous genes, you have a nice network that can be imaged at quite good spatial and temporal resolutions, while the animals do not show any difference in vitality and also in tissue morphogenesis, as we've shown. And just let me jump to this image here, because we are quite excited about this. Nobody knows where and how intermediate filaments are formed, and what drives the formation of intermediate filaments. And this keratin is actually one of the first ones, if not the first one, keratin polypeptide that's made during embryogenesis. So what you're witnessing here in this case, in this time-lapse series of an 8-cell embryo, you see the birth, the de novo formation of keratins in this system here, and you see first this diffuse fluorescence, biosynthetic material, probably, we don't know this, but we can check this now, tetramers, which will then sort of accumulate at cell-cell junctions, forming these more or less punctate petals. This is later time points, and you have in the cytoplasm these little kind of rod-shaped particles, which again show a high degree of dynamics and eventually will form a network which, and I think these cells, yeah, they divide now, become much more pronounced after mitosis. And what is sort of almost a surprise, but quite gratifying to us, is that these punctate particles here correspond to the adhesion sites that are specific for the keratin intermediate filaments that are referred to as desmosomes. And finally, these punctae are also dynamic and not static, like we see it in cultured cells. Let me quickly summarize. I hope I was able to show you that keratins are obligatory heteropolymers on the dimer level. They're nonpolar at the tetramer level. They're assembly competent without any cofactors. They are subject to multiple post-translational modifications, not only phosphorylations. In cultured cells, they are very motile, and this motility depends on the actin and tubulin-based cytoskeleton. They're subject to continuous turnover, but are stabilized upon anchorage to the nucleus and cell-cell adhesion or cell extracellular matrix adhesion sites, and this is regulated by growth factors or stress and mitosis through various signaling pathways. In vital organisms or viable tissues, they are arranged in an evolutionary conserved pattern, contributing to epithelial barrier function, and also dynamic, and their assembly is coordinated by distinct adhesion sites. Now, challenges that maybe some of you can help. We would like to not only experimentally, but also computationally validate and model the keratin cycle in order to predict perturbations of the cycle. We'd like to correlate the, in some degrees, similar but also different behavior of the keratin system in vitro and in vivo, and like to develop a physical model of the intestinal cytoskeleton in silience, because this is a very invariable, it's a very defined system, and would like to examine the keratin plasticity in developing epithelia that is in the mouse embryo. And we do this because we believe that these dynamics of the keratin systems are tightly linked to epithelial functions. And in order to do so, let me just remind that we are talking about different scales. We started with the in vitro assembly, which will, under certain circumstances, can also be induced to form networks about the cellular level, the tissue and organismal level. We're talking about not only length scales, but also time scales in these various dimensions. And being a medical doctor, we are of course interested what goes wrong, what goes astray, to actually exhaust this system and then resulting in disease and what kind of consequences it has at these different levels. And finally, I'd like to use a scheme that I prepared together with Michael Bay a number of years ago, where we stand. So we do have information, not in our laboratory, but many other laboratories, on in vitro assembly behavior from the polypeptide to the network level. We have some information on the in vivo level, from cell to organism. We have information on structure, kinetics, mechanics, and function. There's a large amount of very poorly sorted data, multidimensional data, and we'd like to use this kind of data to extract important parameters that can be used in silico for actually modeling, which can then be used for validation, and eventually also, of course, for simulation to then induce certain types of stresses and predict what kind of consequences it will have at these different levels. And finally, I'd like to thank some of the people that have contributed, foremost Reinhard Windhofer, the keratin cycling engineer, and various other people in the lab who either worked on the C. elegans system, on the mouse, or on the cell culture system and technical assistant, and of course many more. And in particular, I'm grateful to a very fruitful and collaboration with Rudolf Merkel and his laboratories and other groups that we've worked together in the past. I'm sorry I kind of didn't watch the time so much, and I very much thank for your attention.
Thank you very much, Rudolf. I think we should restrict questions to maybe 3 or 4 quick ones.
So what is the persistence length of a single filament?
You always ask me, I think it's a micrometer or something.
Below.
as far as I know, it's below a micron.
Yeah.
Some hundreds of nanometers.
Okay.
Hundreds of nanometers.
Yeah, it's very different from the other ones.
So it's relatively flexible.
Yes, it's like a rubber band. Yeah.
Flexible.
I mean, you see a lot of bends in your images, but then they're on the same scale like this.
You mentioned that someone had knocked down the keratins in a keratocyte. How does the motility change?
Becomes actually faster. It's increased. Till it's increased. And this is also very perturbing, disturbing, because if you do this with other intermediate filaments, for example, vimentin, you may have the opposite effect. So there's something about these filaments makes them very specific, adapted to a given cell type, and they may have very different properties depending on their variable domains in the end.
So is their primary purpose to sort of maintain structural stability and make, when you knock them out, you get a more readily movable cell, or what do you think?
At the surface, yes. I think it comes probably a bit more complicated. You also may need a stabilizing element for migration, right? You need the counterbalance to when you produce forces in a cell. So I think the best answer, at least that's always the answer I give, is nature has tried this a lot, and if you disturb the system even by single point mutations and intervene with proper assembly by shortening, for example, the efficiency of this cycle, the consequence is blister formation upon mechanical trauma. So this shows this is important for mechanical stability in the organism, in our skin. Yeah.
Probably we need to stop here. Thanks very much again. Lunch will be in the working part, so when you